try to read once. All right. If you can get to read once, then you'll be able to understand the pace. So that's what. So before you join the class or you have got 1 week, please make a time and see that you get to read a little bit about the topic, so that when I'm explaining, it will be easier also for you to understand it, because we have got a lot of portion to cover and we have only 12 hours left. And that's what 12, that's what, for every unit they give only 1 hour for the class. So I'll be going slightly fast. That's about it. All right, so let us just go with the this corals belong to the class Anthozoa in the phylum Coelenterates again, and they're actually deposits of lime or calcium, and they are secreted by the polyps. The polyp is a sessile form that is belonging to the phylum Coelenterata. So the polyps secrete these corals, which are deposits of lime or calcium carbonate. Lime is basically calcium carbonate. So some coral polyps are solitary, some of them are colonial in nature, and they are extensively formed of calcium carbonates. All mass is present, so they are known as coral reefs. So again, the polyp structure has been given over here for you. You can get to see that it has this is attached to the. Please, I request you all to mute yourselves. Right. So please mute yourselves. That's about it I'm asking you. So right. So this, as you see, so basically the polyp structure has 3 basic tissue layers with a gastrovascular cavity, which is lined with flagella, helps in the filtration of the nutrients. Then these contain the nidae or the stinging cells, and the body arrangement is radially symmetrical from the outside. But then they are all biradially symmetrical, as we have read about them yesterday. Now you have different kinds of reefs over here. Coral reefs, you have platform reefs, fringing reefs, and you have one more thing is barrier reefs, atolls, etc. We'll come to them later. So before that, when you get to see, this is the coral cup. You can see in your textbook also the transfer section. So you have muscles, you have primary mesentery, secondary mesentery, and you have a theca, and this is the scleroseptum. You can see the septa in the diagram also that is given. So these are the columella and as you see, this middle part is known as the columella, and these are the septum. So a coral polyp secretes a small cup like calcareous skeletal structure, which is known as a coralite or callus. This supports or gives support and protection for the soft-bodied polyp. Now, this coral, individual polyps, all of them fuse together. They form one huge mass, which is known as corallium. And all the polyps secrete calcium carbonate continuously. So what happens is the colonies expand upwards and outwards. And once all the polyps of a coral colony, they are again interconnected with the tissue that is there inside, forming a thin skin over the dead corallium, which completely covers it. So that is the basic structure of a coral or a coral reef that is being formed. Okay, so now the typical coralite, this is a coralite. It has a disc, cup or conical form in nature, measures around 5 centimeters to 25 centimeters. Peripheral wall of the cup surrounds the aboral portion, and that is known as a theca, which is made up of dense stony calcium carbonate. Then inside that you have living spaces which are known as cenoenchyme. Cenoenchyme. Then you have the basal plate, again basic structure, all of them, and then you have the sclerosepta. These are the sclerosepta, as you can see, and then you have the disepimens. So what happens is all of this is known as the columella. Many coral secondary structure are also formed, and this is again like a coralite only. Now, so you have a transverse structure or of this coralite that is given in your book. Please follow that. So you can see in the vertical structure, the first one is the mouth, which is at the that hollow part, whatever. This is the mouth. And then these are the tentacles that are present. And then inside this, you have the epidermis and the gastrodermis, mesoglia. All of it is that has been mentioned. It's a beautiful diagram. It is given in your book. And yes, you just always follow a diagram. You will get half the information. When you are drawing a diagram and along with it labeling, you can mention the same thing, and when you're writing also, and you will definitely be able to get marks if necessary, whenever necessary. Again, in a living colony, polyps occur in, they occupy the upper surface of this corallium. Corallium is the whole mass that is present, the skeletal mass with individual polyps that are fused together. This colony grows in number by budding of the polyps. So it is asexual reproduction that is taking place, as you have already learned yesterday. And then the size and the thickness of the colony also increases whenever the budding takes place. So newly formed polyps secrete again their own corallites. So what happens is the whole thing structure increases in size. Now, two types of corals are present, basically based on their structure. So that is, one is aporose or imperforate corals, and they are again solid and stony. As you know, coral color is usually red to deep red to light red, pink. Different colors are there, usually reddish in color. And then you have even perforate corals with perforations, as in Madrepora. So a lot of examples are given regarding the corals. So examples are, if you see, the hydrozoan corals are Millipora, Stylaster. Then anthozoan corals include the Tubipora, which is the organ pipe coral, Alconium, Alcyonium, then Heliopora, which is the blue coral, Gorgonia, sea fans, and Corallium, which is the red coral. So the red coral, whatever coral or what do you call it, that coral that you get, it is actually the red coral, from the red coral. Then the flabellum, which is a solitary one. Then fungia, astrea. You have so many. Meandrina, which is the brain coral, and the black coral, etc. Please just give me 1 minute. I'll just come. I'll have to. Yes. So when it comes to the coral reefs, you have different kinds of coral reefs that are present. So again, continuous colonies are growing because of the budding of the polyps. So they form extensive masses, which are known as the coral reefs. So according to one scientist who's known as the Wayland Vaughan, a coral reef can be defined as a ridge or a mound of limestone, upper surface of which is near the surface of the sea, and it is formed of calcium carbonate. This is again by the action of organisms, chiefly corals. So again, you need warm, shallow, clear waters, and you have to have the temperate, tropical to temperate climate, which is around 29 degrees Celsius to 33 degrees Celsius. One of the best examples for us regarding the coral reefs is the Andaman and Nicobar Islands. In fact, if at all, you get the opportunity to go over there, you can actually see the coral reefs. You can go snorkeling to see the corals present over there. There are certain islands over there, basically, wherein if you go there, you can see, for example, it's the Neil Island is one, and you have the Ross Island. So once you go over there, you get to see it, like, different kinds of corals, how they're formed, the structure of it, how the calcium carbonate are there, and some of them are of different colors as well. So that's what. Make yourself some time, go for, that's one of the nearest places that we can visit to watch or to see the coral reefs. Anyway, coming back to our lesson, that is, these coral reefs or corals are restricted to tropical waters. Generally, they are found in two regions. One is the Caribbean waters, that is near Florida, Bermuda, and Bahamas, West Indies. All of these are the Caribbean islands and the Caribbean waters. Second is the Indo-Pacific waters, that is from the east coast of Africa through the Indian Ocean. Our Andaman is located in Bay of Bengal, as you all know. And then the biggest coral reef that you can find anywhere in the world is your Great Barrier Reef in Australia, in fact. So, and again, we'll come back to that. And again, what do you call it? The Western Pacific, that is as far as the Hawaiian Islands. And The Pacific northeast of Australia is known as the Coral Sea. Coral reefs are absent in the western shores of Africa and America because of the cold currents. The water over there is cold. In general, a reef extends from the shore to some distance into the sea. Edge of the reef moves away from the shore, is exposed to the tide. So what happens is, as you can see, it is formed from the base to the top. So whatever you're seeing at the top is not the only part. It has to be attached to the bottom, like, attached to the surface of the or the seabed, basically. So again, beyond this, you have the sea, basically. It's known as the reef front or a reef edge. And then they grow into a slope as such. And then the space between the reef edge and the shore is slightly lower. And this is known as this reef flat or the seaward flat. Again, it is depending upon the width and the depth of the particular area that we are talking about. So it can be called as a lagoon. So when it comes to the different kinds of coral reefs, you have the fringing, three major types. One is the fringing reefs, the barrier reefs, and the atolls. So in the fringing reefs, again, you have platform reefs, then you have these are the fringing reefs. So they are on most of the tropical islands, with a few of the coral animals that are present, and they can tolerate this nutrient poor surfaces also. And they are the simplest of the reefs, basically. So with shallow waters up to around 400 meters, which is still shallow according to the other depths that are encountered everywhere else. So this reef edge, whatever, as you can see, this is the fringing reef. So this reef edge, whatever you see, is shallow, navigable for small boats, exposed to low tides. So composed of coral sand, mud, living coral colonies, etc. So this is kind of common in the, the East Indies. Now, when it comes to the barrier reefs, we'll see this is the, you can see this is the island, this is the fringing reef, this is the coral reef. No lagoons or whatever present. These are the barrier reefs. Now, in this, what happens is the seaward, it is flat in nature. Then they growing, so they extend up to 800 meters to several kilometers away from the seaward flat. Then the stretch of water separating the reef will be around 10 to 50 fathoms deep. So they are basically talking about the depth. So you can also call them as lagoons. So this is the Great Barrier Reef as seen in the picture. Along the northeast coast of Australia, it is around 150 kilometers long. And this is the best example. And the reef front slopes to a depth of around 70 to 90 meters down. And that's what, this is around 200 kilometers long and up to 150 kilometers from the shore. That's what. So the length is around 2,000 kilometers long. And from the shore, they're talking about the, the length is around 150 kilometers. When it comes to atoll, these are all small coral islands. See, and it is also known as a lagoon island. So you can see it is more or less circular in nature, oval or horseshoe-shaped reef. Now this encloses the lagoon, and this can be up to around 80 to 90 kilometers across, and there is no land as such. So it's a ring-shaped coral reef. When an island is enclosed by this barrier reefs, then this gets sunken. So it is circular, and this encloses a lagoon, as you can see. And then on the top of the underwater mountains formed by volcanoes. So these are the major ones about, types of coral reefs. And when you see the location, you can get to see that 90%, around 92% are located in the Indo-Pacific region only. This is the Indo-Pacific region. And there are no coral reefs in the west coast of US and Africa as such. That is because of all this area, you have the Atlantic Ocean, the Pacific Ocean, and you get to see that it's all very cold as such. So when it comes to the, this is the largest coral reef, and the Belize Barrier Reef is the second largest. Red Sea Coral Reef is located around, off the coast of Saudi Arabia and Egypt. So see such colorful pictures. In fact, if you get an opportunity to do snorkeling, Whenever possible in your lifetime, you should go ahead with it. Again, one thing is
Voice.
to touch a
Here, madam.
coral. Yes. Yes, please tell me.
Some disturbance, madam.
Yes, somebody has unmuted themselves. I request you all to mute yourselves so that you can listen to me. We are lagging behind in time, and today also I will have to be able to, I will be taking around 20 minutes to half an hour more of your class after 5 o'clock. So please be ready with that. All right. Anyway, so the thing is, we are almost done with the coral reefs. Please, you should not touch the coral reef with your hand, otherwise it will die. It is as sensitive as that. So when it comes to the economic importance of coral reefs, you can see that, on the theory about the coral reef formation, basically, they form very slowly. We are talking about 10 to 200 millimeters. That is 10 millimeters means 1 centimeter to 20 centimeters in a year. So the existing reefs, whatever you see, the Great Barrier Reefs, whatever you see, they have been formed around 30,000 years ago. It is not every day they grow off like that. No, it is not going to take place. So, and they're extremely sensitive to light and extremely sensitive organisms, because of which even if you touch them, they are bound to die. That is why the Great Barrier Reef almost died. But thanks to this pandemic, one of the major advantages of what had happened in the last 2 years of pandemic is that the Great Barrier Reef could somewhat become a little bit better and started becoming or turning green. Otherwise, all the corals were dying thanks to the pollution that was occurring. So anyway, when it comes to the economic importance as such, they're beneficial because they protect the shores from erosion by tidal waves. And then, whatever the reefs, they take it out. In fact, they carve stones out of which they're used for construction building. So we don't exactly need that. We have, and then, this coral reefs serve as habitats for many plants and animals, and they have they are highly prized because they look very good. This is one of the pictures of a sea fan, if I'm not wrong, the red one. So they look beautiful. So they need calcium to build the limestone skeletons and all that. They help in giving a lot of food, shelter to different kinds of organisms. They protect the coasts from strong currents, waves. So that is why it's known as a barrier reef as such. Homes to different sea creatures, and they help in the local economies because tourism, diving, fishing trips, etc. They all help. So they help in symbiosis also. Coral polyps reduce, what do you call it? They help in symbiosis purpose. Basically, algae live in the corals, right? So this is going to be helpful for the algae. Clownfish and sea anemone is one of the most famous examples. In fact, Finding Nemo, if you have seen that movie also, you can get to see that the clownfish are protected by the sea anemone's stinging tentacles, and the clownfish chases away the sea anemone's predators. So again, this clownfish provides the fertilizer, so for the sea anemone again. So food is being produced. And then crabs and corals. So what happens is the crab eats the coral tissue and mucus. At the same time, the coral provides protection for the crab. So it's a symbiotic relationship again. And they're again cleaning stations because what happens is tiny fish feed off the fungal growths. And as such, this is cleaning stations are present in the reef environments. So you have a lot of animals that are present on the coral reefs. Different types of animals include the sponges, the fishes, the crustaceans, snails, mollusks, which rely on the corals for food and shelter. And sponges, we have already seen them. They grow almost up to 6 feet in diameter. So the primitive ones, in fact, live attached to the surfaces. Again, more sponges. These are the cnidarians. We have just seen about them in the last class. So I will be sharing this particular PowerPoint with you, and yesterday's class PowerPoint also. You can just have a look. Now I will be going along with the
Thank you, ma'am.
Yes. Now I'll start with phylum Platyhelminthes. All right, I'm going to stop sharing this. You can see this once you have some free time. Now, what I'll do is I'll start with my Platyhelminthes. There are 2 or 3 PowerPoints. All your PowerPoints, whatever I'm going to share, they're all going to be like, only for you to understand in a easier way. Whatever it is for the notes part, please follow your textbook. And that's about it. Okay. So when we start with phylum Platyhelminthes, we are going to look at the general characters, classification, certain important trematodes, cestodes. Most of them are parasites in nature. And then we are going to study about Fasciola Hepatica, which is the liver fluke. And then we are going to study about the different kinds of larvae that are also present in it. Then we are going to study the cestodes. Cestode parasites against cestodes is one class in Platyhelminthes, which is exclusively parasitic in nature. All of them are parasites. So in this, we are going to have a type study. We are going to see about Taenia solium, which is the pork tapeworm. And then after this, we're just going to see the basic parameters of this particular phylum. So when it comes to that, how is it affecting the how are these parasites affecting the human beings, at the same time the different kinds of domestic animals which are the carriers of them. So phylum Platyhelminthes, which is has very primitive animals, they show definite metazoan organization. So till Cnidaria, we have seen that the body wall has only 2 layers. One is ectoderm and endoderm. You have a amoeboid or gelatinous mesoglia in between, but you do not have a particular layer as such, which is known as the mesoderm. But in Platyhelminthes, you have 3 layers: ectoderm, endoderm, and mesoderm. We'll get to that. So they are free living or parasitic. They're bilaterally symmetrical with 3 germ layers. That is why they're known as triploblastic. So you have an ectodermis, mesodermis, and endodermis. So epidermis again has a cilia or a cuticle surrounding it. And now, because of the absence of a coelom or the cavity as such, it is known as acoelomy. The phylum Platyhelminthes represents all the flatworms in the kingdom Animalia. Platys means flat. Helminthes means worms. So again, as you see, this is the outer ectoderm. You have the middle mesoderm, which is a complete layer altogether. And then you have the endoderm, which is surrounding the gut. And another feature is that they are all bilaterally symmetrical in nature. So you can cut them longitudinally, and they're all flattened dorsoventrally. That is why they're known as flatworms. All these are either terrestrial or aquatic animals. And then they are either free living or parasitic. We have already seen that. And they're bisexual in nature. That is, they are hermaphrodites. Acoelomate animals because there is no internal cavity. And as such, whatever the cavity is filled with the parenchymatous cells. Again, they have some of them, the parasites especially, to hold to the host body. They have chitinous hooks and suckers for the attachment. And the space is filled with, between all the various organs that are present, it is filled with a connective tissue, which is known as parenchyma. You have a cilia or a cuticle that is present around the on the epidermis, and you have locomotion, which is by cilia in certain larvae and by the contraction of the muscles in different adults. You do not have any kind of special locomotory organs in the adult form, not in the larval form. The larval forms might be free swimming or you have some cilia present, but the adults are all sedentary. They are either, since they mostly live as parasites, they are in the host, they are stuck with the help of the suckers or the hooks. Again, the digestive system is not complete. It only has a mouth, a pharynx, and intestine. There is no anus. In some species, digestive system is completely absent, as in the case of tapeworms. Now, you do not have respiratory or the circulatory systems present, and excretory system has protonephridia of flame cells and excretory canals. We'll get to say about that in some time. Excuse me. The nervous system is primitive again, has a pair of cerebral ganglia or brain and around 1 to 3 pairs, that is around 6 of the longitudinal nerve cords, which with transverse connections in middle. They go longitudinal in the body with transverse connections. They look like a ladder basically. So turbal area is one class wherein sense organs are present. Trematodes, excuse me. I request you all to stop using your, what do you call it, do all this representative art. You can do it later when you're free. I can totally understand you would like to, communicate with your friends. But so whoever has written this hello, please erase it if possible. Right. So the nervous system is primitive, as I told you, but except in the case of class Turbularia, and it is reduced in the parasitic forms. So ocelli and ciliary pits are the sense organs in certain animals. So except in some cases, most or all of the flatworms are hermaphrodites, that is, they're bisexual. And a very peculiar feature of this particular phylum is that you have both male and female reproductive organs differently. Fertilization is again internal. There is possibility of cross-fertilization in class Trematoda. Self-fertilization in cestodes is very common. Cestodes are all parasites in nature, so they cannot exactly move out of the body to have fertilization taking place. So self-fertilization takes place in those particular organisms. Development development may be direct or indirect. Quite a number of larval stages are present. Life cycle can be with one host, or it can be with more than one host. How much do you want to take? See, I have 100 change. So when it comes to the classification of phylum Platyhelminthes, it is like, what do you call it? There are 3 classes according to certain investigators or certain scientists. I'm going to stop this. I'm going to share it again because I'm not very comfortable with whoever has written hello over there. Right. Say bye, mommy. So, sorry.
We can't hear your voice, madam.
No, yeah, that's what I unmuted myself. So yes. So some of them, they classify them into 4 or 5 classes. Animals belonging to this group Monogenia, and Digenia are given the status of a class, but some of them only classify them as 3, that is Turbularia, Trematoda, and Cestodia. So when it comes to that, we in our book, they have given thankfully only 3 classes. We have Turbularia, Trematoda, and Cestodia or Cestoda. So let us just have a look at it. Comes to class Turbularia. I request you all to mute yourselves, please. So these are free living, around ranging from 1 millimeters to 600 millimeters. They are predators or scavengers. So they mostly are nocturnal. That is, they come out during the night time. They either live in shaded or humid locations, such as the leaf litter That is, the fallen leaves, whatever is there, which is damp, basically a little bit of moisture is left in that such kind of places, rotting wood. So such places you have a lot of turbularians living. So since 2 millimeters to 60 millimeters, some of them are microscopic also in nature. And again, the color can be around brown, gray, black. And the land planarians and polyclads, they are slightly colored. And anterior end is differentiated into head. Epidermis is, do I have a picture? If you can see a picture, maybe you can see it. Sorry, do not have a picture. Anyway. So body color is, right. Please give me a minute. I will try to share you a picture, a particular slide with pictures as such. I have got one. Right. So these are basic flatworms that you can see, turbularians as such. So anterior end is differentiated into a head, and epidermis is cellular, partly or wholly ciliated, has crystalline rod-like structures which are known as rhabdites. Alimentary canal consists of a ventral mouth. Ventral means on the underside of this particular thing. So this is the dorsal area, this is the ventral area, if you can imagine it. So the dorsal, if you see my hand, this is the dorsal area, this is the ventral area. The inside, the palm side is the ventral area, this is the dorsal area. So the, mouth is on the ventral side, and you have a protrusible pharynx, intestine, and they're again extensively branched, in fact. Then occasionally a free-swimming larva form is also present. So all of them emerge from an egg or a cocoon, and the juvenile form resembles the adult except in the size. It is small in size. So example of these planarians are Planaria, Dugacia, Temnocephala, etc. These are the ones. A good picture of it is given in your textbook, also representative of Turbularia. If you turn to page number 82, representative figure is 5.1. You can get to see whatever I am sharing right now and whatever you see in the textbook is the same. So when it comes to class Trematoda, you can get to see that in class Trematoda, they are flukes. Most famous examples are the Fasciola Hepatica. We are going to read about it. And when you're going to see about the cystosoma, then Paragoninus and Aspidogaster. All these are the examples of flukes, basically. So the adults are ecto or endoparasites. Ecto means that live on the, or outside the body of the host. Endo means inside the liver or inside the body of the host. Body is elongated. And it can be oval, it can be round, but the body is not segmented. There are no segments as such. And it is either with suckers or hooks so that they can attach themselves to the tissues of the host animal. So you have two different suckers. One is the oral sucker, and then you have the ventral sucker at the posterior end. Epidermis is absent in the adult, but the body is covered with a thick cuticle. Cilia, rhabdites are absent in the adults. And these animals have a well-developed digestive system provided with oral sucker. Intestine is divided into two major branches, and they once again divide and subdivide into fine branches. Then this is you have a very primitive circulatory system present, and the excretory organs are the protonephridia. And then you have a pair of cerebral ganglia, and also you have the longitudinal nerve cord that you can see in the transverse section, basically. So you have a longitudinal nerve cord which goes like this. This is the transverse section. What you're doing is you have to imagine it as a ladder-like form. You're seeing from the beginning of the ladder as such. So again, reproductive system is quite well developed in this particular class, and you have 2 to many testes, but in the female you have only one single ovary. and both the testes and the ovaries are highly branched. And in the ectoparasites, development is direct, while it is indirect, at least with 2 larval stages in the endoparasites. Simple practical reason being, the endoparasites, the larval stages have to come out and it has to go thrive in another organism as such, because of which for survival it has different kinds of larval stages. Ectoparasite development is direct, that is, from the fertilized egg to the adult. So it is direct as such. So when it comes to the life history, all these classes, especially in class Trematoda, it is a complicated life history. You have at least 2 or more hosts. As I told you, the example is Fasciola Hepatica. You're going to read more in detail about it. Then Aspidogaster, Cystosoma, and the Paragonimus. So when it comes to class Cestoda, you can get to see that they're all tapeworms as such. So because they look like flat, ribbon-like creatures. And this is the class Turbularia, as you can see with the eyes and all that. I'll get in a tapeworm structure. Yes. This is how big the tapeworm is. They're all endoparasites of vertebrates. They can live in fishes. They can live in mammals. So from fishes onwards to mammals, they're all endoparasites. Body length is highly variable. It can be only up to 1 mm to about 12 meters. They do not have epidermis, rhabdites, but they have a cilia which is covered with a thick cuticle. There are 3 distinct divisions in the body of a tapeworm. One is the scolex. This is the scolex. You have a neck, and then all of this is known as the strobila. So the body after the neck, this part is the neck region, right? After the body region of the neck is known as the strobila, and it has a large number of segments called as proglottids. Now the proglottids at the anterior end. This is the anterior end. This is the posterior end. So what happens is these are small, narrow, and those at the posterior end are broad, and the number of proglottids, the number of these segments that are present, they can be up from 4 to 4,000. Segmentation of the body is called as metamerism, but then the basic rules of segmentation is that it should be a repetition of all the body parts and all of them should be same. But segmentation in the cestodes is not exactly true segmentation. It is known as pseudometamerism, because in cestodes, what happens is new body segments are added at the anterior end. This is the anterior end. Whereas in the annelids, wherein you have the true segmentation present, the body segments are added from the posterior ends. So that is why class Cestoda exhibits pseudometamerism. And with a few exceptions, anterior part of the body is differentiated into a head-like structure, which is known as the scolex. This is the neck. These are the proglottids. Scolex again is not a true head, as there are no sense organs. There is no mouth, but only an anchoring organ that helps to keep the parasite in position in the tissues of the hosts. I'll see if there is a better, please give me a minute. We'll come back to this again. This is the class Cestoda. Endoparasites, anterior end has scolex with a rostellum. Includes suckers. These hooks can withdraw or extend. So the new segments are added at the anterior end from here onwards, and then you have the mature proglottids, or the gravid proglottids as they are known as, at the posterior end. So oral sucker is present with hooks present on the scolex, helps in anchoring with to the host tissues. Narrow neck is present, and then you have new proglottids are added immediately after the scolex, in fact. And these are the mature proglottids, and these are the gravid proglottids. They are shed off from the body by a process which is known as apolysis. Each proglottid, this particular segment, as you can see, has both male and female organs, a nerve cord, excretory canal with flame cells. So this also supports that segmentation is cestodes is not exactly true metamerism as seen in the annelids. Again, digestive system is absent in tapeworms because there is no need for the tapeworm to have a digestive system. All it does is take nutrients directly from the host. So you do not need a digestive system anymore. It is a total waste of time for them. Excretory system is necessary. So you have a flame cells. They open into canals. They travel along all over the body, in fact. And again, a nervous system, you have a pair of ganglia in the form of ring in the scolex itself, and you have two longitudinal nerve cords that travel all over the body. Then again, there's hermaphrodites. I told you, no, both of them, both the male and the female reproductive organs are present in the same, each proglottid. So again, large number of testes is present and only one single ovary is present. So self-fertilization can take place itself inside this particular proglottid itself. And you have a characteristic oncosphere, which is a six-hooked stage, which is seen only during the development. And life cycle has one or more intermediate hosts. Examples are Taenia, Diphyllobothrium, Echinococcus. So Echinococcus is actually known as the dog tapeworm or the hydrated worm. Taenia solium, Taenia sagina. So you have pork tapeworm, beef tapeworm, etc. So many species are there accordingly. The picture is given in your textbook regarding the same. So we'll come back to this. Now I will tell you about Fasciola Hepatica. So Fasciola Hepatica is the liver fluke. Just give me a moment, I'll have to write. So phylum Platyhelminthes, class Trematoda, order Digenea, subclass Digenea, but given in your book, it is order Digenea, family is Fasciolidae, genus is Fasciola, species is Hepatica. Again, it is an endoparasite that lives in the bile ducts of sheep and goats, worldwide in distribution. And especially wherever there are sheep and cattle, you have this particular flukes, basically liver fluke. And this history was given by a scientist who was whose name is Thomas. And this causes fasciolysis or liver rot. And it causes immense damage to the liver and the bile ducts of the sheep. Also reported in the livers of several animals such as horses, dogs, wild animals. Occasionally in the human liver also, that is because of the consumption of contaminated food. So there are two hosts. The primary host is the sheep, in which the adult flukes are attached to the walls of the bile duct. Secondary host is a small specific gastropod snail, Limnia truncatula. So all the larval stages that are present are taking place in this particular snail, that is Limnia truncatula. Occasionally, you have other species of snails such as Planorbis or Bullinus also, which act as the secondary hosts. Adult is soft, dorsoventrally flattened. You know what? If you follow this particular PowerPoint, most of the points from your textbook are covered actually. So it makes it easy to read. So what I'll do is I'll just follow the PowerPoint for this particular explanation. You can just see they're all in points, so very easy to understand also. Multicellular, eukaryotic, no particular cell wall present, and organs are all internal. Again, dorsoventrally flattened, and it is around leaf-like or dark brown body, and it has a bilateral symmetry, so you can just cut it like this. Measures around 1.8, that is 18 to 30 millimeters. In your book, it's 1.8 to 3 centimeters in length. So the same thing, conversion factor, 0.4 to 1.5 centimeters in width, and the ovaries and the testes are highly branched, leaf-like, and you have the cephalic cone or the oral cone or the head lobe present over here at the anterior end, having small circular aperture, which is the mouth. Circular disc surrounds the mouth, and it is well known as the anterior or the oral sucker, which acts as the suctorial organ. Right. So this is the primary host, the sheep, the liver and the bile duct. The secondary host, wherein the digestive glands have the larvae in it. So when it comes to the anterior or the posterior end, you can see that, the oral sucker is there and the ventral sucker is there. Ventral sucker is also known as the acetabulum. And this helps the worm to attach to the bile ducts by creating a vacuum. So between the two suckers on the ventral side is a small aperture, which is known as the gonopore or the genital aperture. And at the extreme posterior end is a small aperture, which is present over here, which is known as the excretory pore, and there is no anus present. So when it comes to the body wall, you can see that, it consists of the tegument, basement membrane. We'll come to that in some time. Again, this is the appearance and the occurrence, how it looks. This is the limnia truncatula. Basically, the primary host is the sheep. Secondary host is the limnia truncatula. So when it comes to the external features, I have already told you, you have the oral cone or the over here. This is the oral sucker. This is the gonopore. This is the excretory pore. This is the acetabulum or the ventral sucker or the posterior sucker. And it is leaf-like, dorso-ventrally flattened. Length and width is given. Surface is pinkish, margins is brownish or black. So anus is absent. When it comes to the body wall, you have the tegument, you have the basement membrane, and you have the muscle layers and the mesenchyme. So tegument has a syncytium, that is mass of cytoplasm formed with the merging of the cells. So you have mitochondria, endoplasmic reticulum, the vacuoles, and all of these are again present. And it is a very tough and a resistant body wall layer, the cuticle layer made up of scleroprotein, in fact. And the tegument, that is the cuticle, should be thick. It is covering the entire body wall. And you know what? It is thick with the only act, so that it can be able to survive the digestive juices that are released from the body of the host. So immediately beneath the tegument is a thin, delicate basement membrane that is present. And below that, you have the muscle layers, outer circular, middle longitudinal, and the inner is the diagonal muscle layers. And all of these are smooth muscles. Sucker region, you have a bundle of radial fibers also present. Then you have the mesenchyme or the parenchyma. It is a special tissue. It fills all the spaces basically. And it has large intercellular spaces also. It's loosely arranged, irregular, with one or two nuclei. So the functions of it basically is for protection, gaseous exchange, waste excretion. Then you have absorption through the pore canals that are present basically. You've got a good, body wall longitudinal section basically, that as seen in the electron microscope. It's given in your textbook. You can just look at it. So, right. So digested food, waste substances are transported by the parenchyma, and because there is no blood vascular system present, all of this transportation takes place through the parenchyma itself. Again, you have mouth presented at the anterior end, oral sucker, and you have the ovoid pharynx having a small narrow lumen. Then this has the radial muscles with the pharyngeal, that is the pharyngeal glands. Opens into a thin-walled esophagus, and this is lined by, again, single layer of epithelial cells. Opens finally all the way, you have the ceca and the diverticula. These are the ceca or the diverticula of the intestines. And then you have the excretory pore. So there is no anus, because of which this alimentary canal is incomplete. Complete, digestive system is one which starts from mouth and ends at anus. since so mouth, gut, stomach, small intestine, large intestine, rectum, anus. This is how a complete digestive system looks like. So if the alimentary canal or the digestive system is incomplete, it means that there is one part which is not present. It can be the mouth also, it can be the anus also. So when it comes to the Fasciola Hepatica, the feeding and the digestion part, what happens is the liver flukes feed on blood, bile, lymph, tissue pieces. So once they do that, it attaches itself to the wall of the bile duct, and it sucks up all the lymph, bile, and tissue pieces through the oral sucker and the pharynx. So you have again extracellular digestion. The food is sucked into the intestinal ceca with the pumping action of the muscular pharynx. Digestion is extracellular. No complex enzymes are present as such. Digested food travels through the ceca walls, branches, transported to the parenchyma to every corner of the body. So waste is again diffused into the parenchyma. Then it is coming out through the mouth itself. So absorption of the digested food materials takes place through the tegument. And food is stored in the form of glycogen and fats. So when it comes to the respiratory system, you can get to see that it is anaerobic in nature. So because of the presence of free oxygen whenever possible, it can aerobic respiration also can take place. So what happens is glycogen and glucose and glycogen are the chief fuels for respiration. So a little amount of energy is necessary for this particular process. So glycogen after metabolysis. We are talking about the tricarboxylic acid cycle, and then we are talking about all that aerobic and respiratory cycles. You have the TCA, the Krebs cycle, and all that, right? So that is how energy metabolism takes place. You get carbon dioxide, fatty acids. So since it is anaerobic respiration, you get lactic acid. If it was aerobic respiration, can any one of you answer what is the product that comes out? You can unmute and answer if you can. End product of aerobic respiration. Okay. Anyway, carbon dioxide and water and energy. That is what you get. So carbon dioxide again diffuses through the body surface itself. It goes into the host body. Basically, that's about it, what happens. Then you get lactic acid, which is again excreted out through the excretory system, goes into the body of the host. When it comes to the excretory system, you have flame cells or protonephridia. So this is the structure of a flame cell. So this is connected with a complicated system of excretory ducts. So you have excretion and osmoregulation that is taking place. They are all modified mesenchymal cells only. So mesenchymal cells is the characteristic of flatworms. So what happens is, so the flame cells are just modified cells. You have pseudopodia over here. It is attached to the excretory ductule. Terminal capillary, you have the cell lumen over here, the basal granule, cilia, pseudopodia, and this is the nucleus that is present. So the cilia which are present over here, as you can see, they're always constantly flickering. If you see a flame, that flame will always be flickering because it is like, the air is slightly moving it. That is why these are known as the flame cells. So they are the modified mesenchymal cells, irregular bulb-like, thin plastic or elastic with one nucleus, intracellular spaces, pseudopodia-like processes. Then lined by capillary ducts, collecting ducts, twigs, 4 branches. These form the longitudinal excretory canal. Simplest way to explain your textbook basically. You've got the picture of it in your textbook also. You can just go through it. So when it comes to the physiology of excretion, again, you can see that the products of fatty acids, carbon dioxide and ammonia, they diffuse into the flame cells from the mesenchyme, moves the excretory fluid through the various ducts. muscles, the excretory fluid is thrown out from the excretory pore. Then it excretes the nitrogenous wastes. So this is all about the excretion and the physiology of it. When it comes to the nervous system of the liver fluke, you get to see that there's a basic nerve ring or a coral. You have the cerebral ganglion. These are the anterior nerves. And this is the central nerve ring that is present over here, or from which you have the dorsal nerve cord and the ventral nerve cord. And these are the lateral nerve cords that are coming over. And these have peripheral branches from the lateral nerve cord itself. So this is present around the pharynx. A pair of cerebral ganglia are present, dorsolateral in nature. So you have a dorsal and a ventral nerve cord. This is the dorsal, this is the ventral, this is the lateral nerve cord. So there are three pairs of longitudinal nerve cords, like I told you. It is the dorsal, the ventral, and the longitudinal. Simplest example is if you look at what I'm showing you right now. Look at the screen, please. Look at my, instead if you can. So the pencil is the dorsal nerve cord. This is the ventral, the lateral nerve cord. This is the ventral nerve cord. So since the body is dorsoventrally flattened, you can just imagine like this is the thickness of the body. This is how it is. The dorsal, the lateral, and the ventral. This is the ventral. Right? Now, and each of them have peripheral branches. And there are small nerves from the ganglia onwards to the anterior end. And the suckers are supplied by nerves. And they are bipolar in nature. That is, you can distinguish the nerves, the head and the tail of the nerve, basically. So when it comes to the reproductive system of the Fasciola Hepatica, they're all hermaphrodites, and cross-fertilization takes place. Gonads they are very well developed. In fact, in the male and the female genital ducts open into a common chamber, which is known as the genital atrium. It is again present at the anterior end of the body, and it goes out through the genital aperture or the gonopore. This is located ventrally just in front of the acetabulum. So this is the acetabulum, this is the gonopore that is present. So the male reproductive system includes the testes, which are paired in nature, and it is tubular in nature, and highly branched epithelial lining, and they have the spermatozoa in them. So this includes the testes, the vasa differentia or the vas deferens, the seminal vesicle. Spellings are slightly wrong in this particular PPT that I'm showing you, but you can see the correct ones in your textbook, basically. Seminal vesicle. You have the ejaculatory duct. You have the cirrus. This is not mentioned in yes, the cirrus or the penis, and the genital atrium. That is the common chamber in which the male sperms, get into it. Right. So what happens is, testes are a pair of highly ramified, tubular in nature, located along one behind the other at the posterior, somewhere over here, part of the body. Wall of the testis consists of the germinal epithelium. Please give me a minute. Right. So the walls of the testis have this germinal epithelium, in fact, and then they produce a spermatozoa. Now a duct, which is known as the vas deferens, you can see it's here, comes out from the middle of the testis, and then it runs anteriorly and fuses with the acetabulum. See, this is the acetabulum, this is the seminal vesicle, to form a large pear-shaped seminal vesicle. And this stores the sperms. Now you have the ejaculatory duct, in fact, over here, which is convoluted. This extends to form the copulatory organ or the penis. See, this is the cirrus or the penis. And then this opens into the genital atrium by the male genital aperture, and this opens through the common gonopore. So you have a lot of numerous prostrate glands also surrounding the ejaculatory duct. In fact, they open into the ejaculatory duct. And then you have the bags like serous sac that encloses the serous, seminal vesicle, serous, and the prostrate glands. Please give me a minute. Right. So when it comes to the female reproductive system, you can get to see that it consists of a single ovary. You have an oviduct, you have a uterus and associated vitellaria, and you have Malice glands. Special for this particular, the females of the trematodes, in fact. And ovary is highly branched again, anterior to the testes on the right side it is present. Oviduct is present. I think, this picture only will help in both. You can see both of them in this picture only if possible. Female genital aperture is there. So you have an ovary, oviduct, uterus, associated vitellaria. Again, ovary is highly branched, but it is not paired. Only one single ovary is present. Lies on the right side of the body. So somewhere over here, you have the Malice glands, you have the uterus. Please see this picture properly, you'll get it. And then, the walls of the ovary again have germinal epithelium that produces and sheds the ova into the cavity. All of these unite posteriorly to form a short oviduct. You can see that in the picture. See, this is the oviduct that is present over here. These are the ovary. This is the ovary. This is the oviduct. And it runs till it forms a vitelline duct over here. This is the vitelline duct over here. And this again goes and joins from the junction of this oviduct to the vitelline duct. You have a uterus that is present. And this uterus extends up to the genital atrium over here and opens through the female genital aperture. This aperture is close to the male genital aperture on the right side. So it is closed. Both the male and the female genital apertures are closed. And then this uterus stores a large number of capsules called as, which have fertilized eggs. Then the terminal part is muscular, serves to eject the capsules. And this is known as the metra-term. And then during the breeding season, what happens is the short slender tube, which is known as a Laurer's canal, it arises from the terminal, oviduct, runs upwards to open out in the dorsal surface of the body, and this serves as the vagina during the copulation time. Now, there are two important glands associated with the female reproductive system. One is the vitelline glands or vitellaria, and the second is the Malice glands. So vitelline glands, you can see in the textbook, you can see in the PowerPoint itself, these vitelline glands and these Malice glands. The vitelline glands are small, minute, spherical bodies that are scattered around. Though what happens is they form the two longitudinal vitelline ducts. And they are connected with a transverse vitelline duct again. So this enlarges in the center to form the yolk reservoir. From there, what happens is this yolk reservoir produces the yolk cells. The vitelline glands produce the yolk cells that have the albuminous yolk for nourishing the embryos. And then they also have droplets of the shell-forming material. Malice glands are present. They are numerous, unicellular in nature, shell glands that surround the oviduct. In fact, they are open into the junction of the oviduct, median vitelline glands, and the uterus. This secretion of the Malice glands helps in the lubrication of the eggs, so that the eggs can go up to the female genital aperture for that matter. So that, and they also help in activating the spermatozoa, and they also help in hardening the shell, the egg shell. So when it comes to the development, so this is how it is. The follicles, interconnected ducts, two longitudinal ducts, transverse vitelline ducts, median vitelline duct to oviduct, which produces yolk and shell. So when it comes to Fasciola Hepatica's life cycle and development, it is a very complex. Several larval stages are present and two hosts. So it is known as digenetic. Again, let us see how it is. Infection to the primary host, which is the adult. So copulation takes place, fertilization, egg formation, segmentation and early development, larval stages, wherein it is in the snail. So when it comes to this, what happens is it is a rare occurrence self-fertilization in the case of liver fluke, that is Fasciola hepatica, because as it is a hermaphrodite. So it is usually cross-fertilization that is taking place between two liver flukes in the bile duct. They have the genital pores in opposition basically. So because of which, what happens is copulation is made easier. So the cirrus of one fluke, that is the penis of one fluke, penetrates into the Laurer's canal of the other fluke and injects the spermatozoa. That is where the Malice glands and the vitelline duct come into place, wherein the glands of the Malice glands, the secretions help in the activation of the spermatozoa and in hardening the eggshells. So the spermatozoa move to the distal end of the oviduct where the eggs from the ovary reach, and thus fertilization occurs internally. Then the fertilized eggs receive the yolk cells from the vitelline glands. They get enclosed in the chitinous shell by the that is formed by the granules in the yolk cells. Now, the shell hardens, becomes brownish yellow in color, and it has an operculum at one end. Now, the shelled eggs are called as capsules. They pass into the uterus where the development starts. Now, the capsules come out of the uterus through the gonopore into the bile duct of the sheep. They reach the intestine and then they are passed out through the feces of the sheep. Each fluke produces nearly 5 lakh capsules. So, a single infected sheep can dispense 100 million eggs. And so it is so important because of the fact that it has to be able to adapt to survival and continuation of race, because the chances of survival are very limited. So when you see the life history of Fasciola hepatica, you can get to see that adult fluke, we'll come to this later, cleavage and early development. So as you see in cleavage, what happens is in the uterus, it is complete and holoblastic. You have propagatory cells that are produced, which are small. And then you have the somatic cells, which are large. Somatic cell divides and forms the ectodermal tissue. The propagatory cells are small. It has a lot of multiplication the division taking place, of which you have two cells. They form the larval body and the germ cells. So the larval body forms the ectoderm and the mesoderm, and the other part forms the germ cells. Now further development takes place when the capsule come into contact in the damp areas, and optimal temperature for this ranges from around 22 degrees Celsius to 25 degrees Celsius. As you can see, I'm in the 15th slide. We have up to 21 slides. That is around 6 more slides. So please be patient. Then I'll be able to finish this lesson as such. So in this, again, you have 5 different kinds of larval forms over here. You have the Miracidium, Sporosis, Radia, Cercaria, and the Metacercaria. You have the operculum. This is the fertilized egg or zygote. These are the yolk cells which provide nourishment. These are the egg shells as such. This is the propagatory cell. This is the somatic cell. This propagatory cell is small. Somatic cells are big, form the ectodermal layer. So this is the operculum, as you see, this is the embryo after fertilization. These are the yolk cells that are providing nourishment. This forms all the parts of it. Now, under favorable conditions, you get to see the Miracidium larva. It's there in your textbook as well, but this is a more colorful picture. You can see this hatches out of the operculum, minute. It's around 0.07 millimeters. So it is less than 1 millimeter. So it is microscopic in nature almost. It has many cilia attached. You can see the cilia around it. You have a tetraboratum. So again, what happens is the cilia, it differentiates into the cilia within around 4 to 15 days, in fact. Secretes a proteolytic enzyme that dissolves the operculum, and this emerges out into the water. And it has a head lobe or apical papilla. In fact, you have the apical glands, and then on each side of the apical gland you have the penetration glands that is present. On the dorsal side there is a large brain. Please see here. And this brain is there is again, you have around nerve fibers that are present, and two pigmented eye spots are also present. These are the pigmented eye spots that are present. Then you have protonephridia or flame cells that keep coming out of that and opening into the exterior to the excretory pore that is present over here. In the posterior part, you have groups of cells which are known as germ balls. These are the germ balls present. They are the developing embryos, in fact. Now, Miracidium larva does not feed, swims actively in the water, and it goes in search of the snail, that is the Limnia truncata. If it does not reach a snail, it will die within 24 hours. So if it reaches a snail, then it penetrates with the help of the penetration glands into the snail's body by the apical papilla, and then it bores its way into the snail with the help of the penetration glands. Then it casts off the ciliated epidermis, and it goes into the pulmonary sac or the pulmonary chamber of the snail, where it undergoes into the second larval stage, which is known as the sporocyst. This is the sporocyst. In this sporocyst, the eyes are lost, the glands are lost, the brain is lost. Again, in this, what happens is, in this sporocyst form, elongated structure, you have a thick cuticle that gets developed. Body wall has all the layers except the ciliated epidermis. Now, pair of protonephridia are present basically, and then each are on both the sides of the body. Then this divides into the two flame cells, and this comes out with the from the common excretory pore that is present. This is the kind of larva, as you can see. This is the sporocyst. This is the radial larva. So again, many repeated divisions are taking place, and then you get the third type, which is the radial larva. It is named after the scientist Redi. In the year 1620, he was alive during the 16th century, in fact, the 17th century. So each porocyst divides into or produces around 5 to 6 radii. Radial larva emerges out from the sporocyst and ruptures the body wall. This radial larva again is elongated, cylindrical, has a cuticle. In fact, this is the radial larva, as you can see. Anterior end has a mouth, pharynx. There is a small gut. You have muscle bundle surrounding the pharynx, which is so this is known as the collar. In fact, you have behind the collar itself, you have a birth pore, and then you have conical structures which are known as lappets. And then you have loose, they are muscular projections. They are known as lappets or procruscula, present ventrolaterally near the posterior end. And then you have a lot of unicellular pharyngeal glands that open into the pharynx. Protonephridia further divide so that you have a branched excretory system. So there is a common excretory duct near the lappet, and the cavity of the larva is having germ balls and mesenchyme. So what happens is, this is a very active larva. So what happens is, aiding with the help of collar and lappets, a lot of muscular contractions occur. And then during the summertime, the daughter radia are come out, in fact, and they give rise to the morphologically identical, generation of larvae, radii. But in winter season, they produce the fourth, or autumn season, they produce the fourth larval stage, that is the Circadian larva. This leaves the body of the parent through the birth pore into the digestive gland of the host. This host that we are talking about is the host snail, Limnia. So when it comes to the Circaria larva, you can see that it is oval in nature with a tail. It has an oral sucker, a muscular pharynx, the esophagus, with the body divided into two parts, like, the alimentary canal or the gut. And then this is the acetabular or the ventral sucker. These are the germ cells. Excretory vesicle, excretory ducts. You have the excretory pore over here. So again, like we told you, it is an incomplete, what do you call it, digestive system that is present. You have a certain amount of nephriospores, and then you have the digestive, what do you call it, the mature Cercaria migrates from the snail's digestive glands to the pulmonary sac. From there, it escapes into the surrounding water through the pulmonary sac and through the pulmonary aperture. So around 35 to 65 days is necessary for the Cercaria to go out of the snail's pulmonary sac into the outside water. Then the larva swims for around 2 to 3 days and it loses its tail. Then it gets enclosed in the cystogenous glands. This encysted larva is known as the metacercaria. In this form, it can live for many months altogether in moist conditions. So it has a round body, thick, hard cyst, large number of flame cells are present. There is no tail. Cystogenous glands are present. Excretory bladder opens out through a single pore. And no cystogenic glands, and the genital rudiments are, present. So when it comes to the life cycle, you get to see that, when it comes to the, it is very interesting to see that all this Miracidium, Sporosis, Radia, Cercaria, they're all from the germ cells. So they are the products of the propagatory cells. And in the very beginning, after the cleavage, there is only distinction of somatic and the propagatory cells. Now the propagative cells form all the larval stages. The stomatic cells, whatever are present, they produce the larval body. Now, germ cells of the larval body again develop into the next stages of the larva. And at the same time, some of them remain as a stalk. So all of them are not forming one particular thing, and from that again another. But a stalk of germ cells is there, which produces each larval stage. So there is only a single propagatory cell that is present because of the first cleavage that is occurring. Then that produces a succession of larval stages. This gives rise to the somatic cells at each stage, with some being the stalk cells. Now, when it comes to infection to primary host, what happens is this Metacercaria larva, which has that full particular large or a huge cyst for that matter. In the primary host, what happens is through grazing, it goes into the, it enters the digestive tract. It is there in the water, right? So when the sheep eats that, grazes that, whatever is there, what happens is it goes into the digestive tract. The cyst is dissolved. Then you can see this is the Metacercaria eaten by sheep and exists in the digestive tract. Adult liver fluke resides in the liver cell. And again, this eggs containing the Miracidium leaves, the sheep gut through the feces. Miracidium larva develops, hatches in the water. This enters the snail. From the snail, this is aquatic snail limnaeus. Sporosis with developing radiae. Radiae develop into the Cercaria, free swimming form, which loses the tail to form the Metacercaria. This is how it takes place in the host. Primary infection. After this, again, after a day or two, what happens is that's what the cyst wall is removed in the intestinal wall itself. And then it, from that time onwards, takes around 5 to 6 weeks. This causes severe damage to the liver tissue of the host itself. So after entering, this causes liver rot or fasciolysis. The larva now enters the bile duct, where it finally attains the maturity, sexual maturity. And from 11 to 13 weeks inside the liver of the sheep, it starts laying eggs. And you have seen the complete life history. Now, when it comes to the pathogenicity, you can get to see that there is inflammation of the liver cells leads to hepatitis. So since it lives in the gallbladder also, it causes calcification. So gallstones are there, and it is there in acute, especially when it is young sheep, liver rot or fasciolysis. So yes, because of the interrupted blood supply, it causes brain stroke and death of that particular sheep. And after a few weeks of infection itself, you get to see that there is obstructive jaundice, acute anemia, you have irregular rumination, that is, it is not able to eat properly, and there is a fever, increase in the respiratory activity. And after 3 weeks of infection, you can get to see that edema means inflammation or swelling. So there is edema in the jaws, then you have edema in the abdomen and loss of weight, eye sockets become pale, liver is enlarged, and finally the animal dies. Now, if the host survives, this liver fluke can migrate to the duodenum and then it escapes from the host. Liver, then digestive, so it escapes out of the feces. That is what I told you first. So that is done by escaping to the duodenum, then through the fecal matter, it comes out of the host. So now when it comes to the preventive or the control measures, what can you do? You can treat the diseased animals after seeing that symptoms with hexachloroethane or and tetrachloroethane. This destroys most of the flukes. This preventive measures include killing of the immediate host, that is the snails, by sprinkling copper sulfate solution in the ponds and the ditches. And also you can introduce ducks to pastures. So what will happen is ducks eat snails. So this will reduce the snail population. When the snails are killed by draining the pastures, as the snails cannot survive longer periods of drying. So in case you drain out or dry out that particular area with a lot of moisture or water is there, the snails will die. If the snails die, infection to the primary host, which is the liver, which is the sheep, will not take place, because of which liver fluke or Fasciola hepatica cannot enter into the sheep. So this is all about Fasciola hepatica. The next class, that is the next Saturday, I will continue with the cestoda, and I will also tell you or take the class about the Ascalminthus, which is your nematodes. All right. So thank you for your time, and again I apologize for having taken extra half an hour. So you can all leave right now.
Thank you, ma'am.
All right.
Thank
Okay,
you.
ma'am.
Thank you.
Thank you, ma'am.
Ma'am.